So today we're going to be talking about the senses, as Ellie mentioned, and it's by me. And would you guys like to introduce yourselves as well?
Maryam.
Soumya.
Aria.
All right. Okay, so we'd like to start with this video, and Ellie, just give me a thumbs up if you hear it. I think the sound works though. So.
Surely you know the five senses: sight, smell, taste, sound, and touch. But do you know how they actually work? How do these sense organs pick up information from the surroundings and transmit it to our brains? All of the senses have something in common: sensory receptors. Sensory receptors detect changes in the environment. We'll look at 4 of the specialized receptor cell types: mechanoreceptors, chemoreceptors, thermoreceptors, and photoreceptors. Mechanoreceptors respond to forces and movements. Chemoreceptors respond to chemical substances. Thermoreceptors respond to heat, and photoreceptors respond to light. Now let's see where these sensory receptors are put to work in our senses. Olfaction is our sense of smell. The olfactory bulbs of the brain are located on the other side of the skull bone from the nasal passage. When molecules enter our nasal passage, let's say a molecule of vanilla, the vanilla molecule will bind to an olfactory receptor in the nasal epithelium. The olfactory receptor is a type of chemoreceptor because it's activated by specific chemicals or groups of chemicals. The vanilla olfactory receptor is specific to just the vanilla molecule. Once activated, the olfactory receptor sends a signal through neurons to the olfactory bulb in the brain, and then it's sent to be processed in the olfactory cortex. Humans have about 400 different odorant receptors, which allows us to identify around 1 trillion different odors. But other animals like mice have 1,000 different receptors, and by comparison, humans don't have a very sensitive sense of smell. Aquatic animals can also smell dissolved chemicals in water, just as we smell dissolved chemicals in the air. Gustation is our sense of taste. Within the taste buds on our tongues are many gustatory receptor cells with hairs that will bind to the molecules in food and send messages to our brains. Gustatory receptors, like olfactory receptors, are another type of chemoreceptor. Our sense of sight uses rods and cones as the main sensory receptor cells. Rods and cones are located in the retina of the eye, and they're types of photoreceptors. The rods allow you to see in dim light, and the cones detect color and bright light. Our sense of hearing uses complicated structures in the ear. Beyond an almost Rube Goldberg maze of membranes and tiny bones is a snail-like region called the cochlea. Deep in the cochlea are tiny hair cells, which are mechanoreceptors that register movement that's amplified from sound waves that enter the ear. Mechanoreceptors in the semicircular canals are also used to help with our sense of balance. In the skin are many types of receptors, including Merkel cells, which are mechanoreceptors that detect touch, and thermoreceptors, which can sense hot and cold. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter at SciencePet.
All right. Before we move on, I'd just like to mention that we used brainfacts.org, which is like the BrainBee website, for a lot of the information that we used for this presentation. And so we strongly suggest you all also look into it. It's been redesigned as well, and it's much more user-friendly. And I'm pretty sure as 2021 is coming that they're going to find a virtual way to hold competitions. So I strongly suggest you look into it as well. We also have linked the links we used, like other than brainfacts.org. And so if you look at our slideshow presentation, which will be on the website, you can check those out as well. As the video mentioned, these are the basic 5 senses: smell, touch, taste, sight, and sound. And there are more than these like basic senses, including tension, pain, gravity, temperature, etc. All right, so how we receive the information from our senses are the sensory organs.
Okay, so the 5 main topics we're going to cover today include audioception, which is hearing, and then ophthalmoception, which is sight, olfactoception, which is smell, gustoception, which is taste, and tactioception, which is touch. So first, I'm going to go over audioception, and then before that, I'm going to go over a brief explanation of the anatomy of the ear. So there are 3 main parts of the ear: the outer ear, the middle ear, and the inner ear. And the outer ear contains the pinna, and that's where the sound waves will first collect. And then the middle ear contains 3 ossicle bones called the stirrup, anvil, and hammer, and these serve to amplify the sound. And then in the inner ear, we have the oval window and the cochlea, where transduction is going to occur. And transduction is when the sound waves are converted into neural impulses. So this is a more in-depth slide of how audioception works. So first what happens is the pinna or outer ear directs the sound waves into the auditory canal. And at the end of the auditory canal is the eardrum. So once the sound waves reach the eardrum vibrates, and this stimulates the 3 ossicle bones on the other side of the eardrum. And after the eardrum vibrates, it stimulates the ossicle bones, and these ossicle bones amplify the sound even more. And then the oval window is stimulated, and the oval window is right next to the ossicle bones. And then this stimulates the fluid in the cochlea. And when the fluid moves, the hair cells on the basilar membrane also move. And this is where the process of transduction occurs, when the sound waves are converted into neural impulses. And then after that, the neural impulse is sent through the auditory nerve into the thalamus and the auditory cortex of the brain. And then there's also hearing loss, which can occur if some parts of the ear are not functioning properly. And so the 2 main types of hearing loss are conductive hearing loss, which occurs when the ossicles in the middle ear are unable to amplify the signal to the eardrum. So this is when you can't hear something clearly. And in order to fix conductive hearing loss, you probably will need a hearing aid. And then the second type of hearing loss is sensory neural hearing loss. And this occurs when the cochlea does not work. And this makes it so you are unable to interpret sounds. And so for this, you'll need a cochlear implant. And this is a surgical procedure. And after you get a cochlear implant, it will stimulate the auditory nerve to convert the sound waves into neural impulses. And so this is a picture of the hearing aid and the cochlear implant. So this is just showing that the hearing aid is for fixing conductive hearing loss, and the cochlear implant is for fixing sensory neural hearing loss.
Okay, so I'll be talking about ophthalmoception, which is the eye. So ophthalmoception is the ability of our eye to focus and detect images of visible light on photoreceptors in the retina, which then generate electrical nerve impulses for varying colors and brightness. There's 2 main types of photoreceptors, which are rods and cones. Rods are very sensitive to light, but they don't distinguish color, but cones distinguish color. So we're going to start off with some of the anatomy of the eye. The iris uses muscles to control the amount of light that enters your eye. So depending on that, it constricts or dilates. The cornea functions as the window to control and focus the entry of light, and it contributes about 75% of the eye's total focusing power. The retina receives the light that the lens has focused and converts it into neural signals, which are then sent to the brain for visual recognition. And our retina has about 120 million rod cells and 6 million cone cells. The lens passes through this watery fluid called the aqueous humor, and it enters the pupil. And the lens can basically bend light and it'll focus it back onto the retina. Then we have two types of fluids known as the aqueous humor and the vitreous humor. These basically maintain pressure within the eye, and since the lens and the cornea have no blood supply, it performs the job of carrying nutrients. And then we have the sclera, which is the tough protective covering around the eye, and it reduces friction between your eyelid and the cornea when you blink. And lastly, we have the optic nerve, which transmits visual information from the retina to the brain. And here I have 4 steps of how light travels through the eye. It first passes through the cornea into the lens. Then the cornea and lens help focus the light rays onto the retina. Then the retina absorbs and converts the light into electrical impulses, and these impulses are transferred along the optic nerve to the brain. So how does our eye play a major role in stimulating a response in other body systems? While our eyes perceive information and send the signals to our brain that triggers responses in other body systems. And the ability of our eyes to send signals to our central and peripheral nervous systems is what keeps us alive and allows our reflexes to work within seconds. And these are some examples. For example, when you watch a scary movie, your heart rate and blood pressure increase, which affect your cardiovascular system. And then if you're in dangerous situations, you can release adrenaline, affecting your endocrine system. Next is tactioception, which is the body's ability to feel physical sensations. And these senses use several modalities like pressure, skin, stretch, vibration, and temperature to transmit messages to the brain. We're going to start off with anatomy of the skin as well. The skin is made up of 3 layers: the epidermis, the dermis, and the hypodermis. The epidermis in this picture is shown, and it's made up of stratified squamous epithelial tissue that protects us from wear and tear and acts as a waterproof barrier. We also have the dermis, which is where most of our nerve endings and glands are located, and your sensory receptors are located there as well. So if you touch something hot, your nerves in your skin shoot messages of pain to your brain, and then the brain sends messages back to the muscles telling you to pull your hand away from whatever it is that's hot. Then the hypodermis is made up of fat, also known as adipose tissue. So the 2 main sensory receptors found in skin are known as thermoreceptors, which is temperature, and then nociceptors, which are pain receptors.
Sorry. All right. So we're moving on to the nose. The nose signals to the brain through the olfactory epithelium, which holds millions of sensory neurons. So when an odor molecule enters the nose, it has a really strong fit with a certain type of receptor. And once it finds its fit, the receptor gets activated. And olfactory receptors are the proteins at the tip of the cells, and they're the ones that bind to the odor molecules and get activated by them. So here's like a step-by-step process on the different parts of the brain that reacts to an odor molecule. So an odor molecule binds to a receptor and it starts an electrical signal from sensory neurons to the olfactory bulb. And the olfactory bulb is the one that relays the signal to all the other parts of the brain for additional processing. So here are some examples of other parts of the brain that aren't directly involved with the smell, but they still process the information. So for example, piriform cortex is a bunch of neurons behind the olfactory bulb, and they're the ones that identify the smell. The thalamus is what helps spread the sensory information. So it's kind of like the olfactory bulb in the fact that it's the one that relays info to the orbitofrontal cortex. Now, the orbitofrontal cortex is the one that helps mix the smell information with the taste information. And in case you don't know, taste is reliant on smell, and you can't taste food without the sense of smell. And a fun fact that scientists have found out is that smell also triggers emotions slash memories because the thalamus also sends information to the hippocampus and amygdala, which mediate learning and memory in the brain. And here are two diagrams. The first one is just how the nose and brain are interrelated and how the odor molecule gets relayed to the brain. While the second image also connects the nose system with how taste works as well. And you can see how they're related to each other.
Okay, so I'm going to be talking about tongue and the taste, which is gustioception. So first, taste buds, which are also called gustatory caliculi, are near gustatory papillae, which I'll go into a little more in a minute. And an adult or average person has around 2,000 to 10,000, and it varies very differently between somebody's age. So basically what happens is you have chemical reception based on papillae. And these are raised bumps on the tongue that can or cannot contain taste buds. And I'll go over into the different types. So when you taste, the taste buds get activated and the brain gets informed on what tastes were there and the level of each taste. Along with this, papillae have microvilli, which are sensitive microscopic hairs, which help with taste perception. And so a sensory cell lifespan is around 10 days, but it doesn't get replaced on a one-to-one basis. So that's how over time, like you have less taste buds and they deteriorate with age. And then there are 4 types of papillae: circumvoltae, fungiform, foliate, and filiform, and each are different. So first, the circumvoltae, these are the largest papillae. They're in the back of the tongue. They contain taste buds and they're in a V shape. Fungiform, they're at the tip of your tongue. They are shaped like mushrooms and they have taste buds and sensory cells. Foliate, they're on the sides of your tongue. They have taste buds and they're leaf shaped. And then filiform, they're round and small, but they don't contain taste buds. So they're actually for chewing and speaking and cleansing of the mouth. So next, the types of taste. So the taste buds can taste 5 different types. They can taste sweet, salty, bitter, or umami. So umami is actually Japanese for savory, but it's like the meaty taste that you have in meats and broths. And then, like it says here, a lot of people think that you can only taste like some flavor, some taste on different parts of the tongue, but in reality, you can taste all the taste across the whole entire tongue. And they sense taste, not flavor. So for instance, with hot, like not temperature hot, but it's actually like the nerve endings rather than taste buds. And like Neha says, the nose plays a very large role in building flavor. And I put this little diagram on the side and it shows like the different types of proteins that are picked up. Or for instance, salt has a sodium channel. And for instance, umami picks up on glutamic acid, and sour is like H plus ions and salt is Na. So they all pick up on very different things. And then lastly, I just wanted to go over taste in the brain. Wait, Neha, can you change it? Yeah, thanks. So basically what happens is food, you eat food and then the microvilli and papillae and the taste buds all interact to get a signal, and then it goes through the gustatory nerve into the cranial nerves 8, 9, and 10. So if you didn't know, there's actually 12 cranial nerves, and they actually have a lot to do with the senses, but the gustatory system is 8, 9, and 10. And then this goes to the thalamus, and then to the gustatory cortex where the information is processed.
All right, so that's the end of our presentation. Hope you learned something. And while we were researching for this presentation, we came across this link from brainfacts.org, I believe. And the link is here, and you can actually get a 3D view of the brain and click the different parts of the brain to see what the function is. So highly recommend you check it out. And that's the end of our presentation.